Thank you very much. That was a superb presentation. We're sorry you're not here in person. We're now going to have Duane's presentation, but it will be given by Dr. Scott Weaver. Thank you.
So we'll move through Dr. Gubler's material fairly quickly because we're running a little behind. So arboviral diseases known to be transmitted by this particular subgenus we keep referring to, Aedes stegomyia. You've heard all of these except epidemic polyarthritis, which is caused by Ross River virus in Australia and the South Pacific. So these are some of the viruses that we may want to be paying attention to, either based on historic information that you've just heard about, or what we suspect to be certain characteristics that may lend themselves to emergence. And I'm just going to mention a few of these. Anyang-yang is a close relative of chikungunya found in Africa, produces essentially the same disease as Mayaro, Ross River, and chikungunya, but it's Anopheles-borne. This is Duane, one of the closest relatives of Zika virus. We know very little about this virus at all, other than it's been associated with human disease. Japanese encephalitis, another closely related flavivirus, much better understood than Zika, can cause severe disease and is very common in Southeast Asia. And then Rift Valley fever virus, this is a Bunya virus that occurs all over sub-Saharan Africa, occasionally causes major outbreaks in domesticated livestock and spill over to people. There's reason to believe that this virus could establish local transmission here in the United States. We already know that many of our mosquito vectors are competent to transmit this virus. And then a couple others like Sinbus that produce fairly mild diseases. So this is an avian alpha virus that also could easily be spread to other parts of the world beyond Europe and Africa where it's known to occur. Back to these urban mosquito vectors, why are they good vectors? And especially, we're going to talk here about Aedes aegypti. There are certain properties of its ecology and behavior that make Aedes aegypti the perfect vector of these viruses. It tends to bite during the daytime, but more importantly, it preferentially bites people. Even when it has the opportunity to bite domesticated animals, it tends to bite people preferentially. It's highly domesticated. The larvae are found in containers of various kinds in people's backyards, in water storage containers in many of the parts of the world where there's no domestic water service. And the adult females tend to enter homes and stay there for the duration of their life. So they have extremely high levels of exposure to people, and they also tend to bite more than once during a reproductive cycle. They may bite several people in the same household, increasing their chance of infection and also transmitting to multiple people. Another vector, Aedes albopictus, its behavior is not quite as conducive to transmission as aegypti. It's also a day-biting mosquito, but it has a more Catholic preference for different kinds of hosts. And it also tends to be paradomestic, but also feral. You can find the larvae both in natural and domestic water containers. So these potential vectors are two of several others believed to be involved in some of the recent outbreaks. In the South Pacific, Aedes polynesiensis, in Yap, also Aedes henselae, Aedes malaysiensis, and other Aedes scutularis species have probably been involved in different outbreaks on different islands. In the US, if Zika arrives here and begins circulating, what other mosquitoes besides Aegypti and Albopictus could be involved? Aedes tricereatus is one well-known vector that transmits a virus called the Crosse virus. That's a major cause of pediatric encephalitis here in the United States. What about other potential mosquito vectors? Well, as shown already, many different species have been implicated as being infected in Africa, but we don't know which of these have the capability to transmit efficiently. But they include many different genera such as Culex, Anopheles, Mansonia, and Erythromatopodes mosquitoes. Traditional methods have basically failed us for dengue, more recently chikungunya, and historically also for yellow fever when we had major urban epidemics here in the Americas about a century ago and earlier. So space spraying, what you typically see in the United States is a pickup truck driving up and down the streets spraying an aerosol of malathion out the back. That's very ineffective for controlling Aedes aegypti because, as I just mentioned, the adult females stay inside of houses, and those sprays don't penetrate very well into houses. Perifocal control around cases could work well for controlling the initiation of the cycle after an importation. If we could quickly diagnose and identify these people, we could do very limited spraying in their house and nearby houses to control Aedes aegypti because it has a very limited flight range, only about 100 meters over the course of its lifetime. Targeted source reduction, if we eliminate the larval habitats of Aedes aegypti around houses, sometimes in sewers or other locations, we can have a major impact on reducing their populations and in suppressing transmission. And then integrated vector management involving not only source reduction but targeted control using different methods than we typically use to control vectors of West Nile, for example, which are outdoor biting, nighttime biting Culex mosquitoes. And it's really critical in these kind of interventions that the community participate. Communities and even individual homeowners have to take personal responsibility for eliminating Aedes aegypti from their property. There are also some bio and genetic control approaches that I'll mention in a moment. So this is typically how we control mosquitoes here in the U.S. for something like West Nile, almost completely ineffective for Aedes aegypti control. What's our history of controlling Aedes aegypti, especially in the Americas? Well, there have been some notable successes in the past, including in Brazil. And as part of a global malaria eradication campaign, there are sort of renewed efforts to find better ways to control mosquitoes. But for Aedes aegypti specifically, there was a hemispheric eradication program implemented for about 2 decades, from the 1950s to the 1970s, as part of a yellow fever eradication program. And Aedes aegypti was eradicated from almost all the countries in the Western Hemisphere. You can see that in 1970, only 3 countries in South America, some of the Caribbean islands, and the southern U.S. still had Aedes aegypti present. But unfortunately, these efforts waned, and Aedes aegypti has now returned to almost the identical distribution we saw in the 1930s. So why did this effort succeed for a couple of decades? Well, first of all, there was a top-down approach in many locations because there were paramilitary governments in place that could exert a strong arm and make sure there was very good compliance in communities. There were dedicated, disciplined staff available to implement these programs. Our cities in the neotropics were much smaller in those days, and there was a lot of detailed mapping to identify the sources of the larvae for Aedes aegypti and to eliminate them. We had fewer automobiles, so not as many used tires, far fewer plastic containers. I think this is a major factor in Latin America. When I started working there about 30 years ago, all the beverages were sold in reusable containers with a deposit on them. Now everything is disposable plastic, and even a small bottle cap is enough for Aedes aegypti larvae to develop. Economic development has taken precedent over many of these activities. There hasn't been adequate funding for these programs, or rather there was adequate funding earlier, not so much today for these programs. Another important factor that in those days we used residual insecticides like DDT that in most places are now not considered environmentally acceptable. So why did we fail after this program ended? Well, the major drivers were urban growth in these tropical locations, the lack of effective control, and then globalization continues to move viruses, people, and mosquitoes around the world. Aedes albopictus, for example, was introduced in the United States just 30 years ago. The outdoor space spraying, our mainstay control method here in the U.S., doesn't work for Aegypti, and the community-based programs on their own have not been effective and sustainable. So the lessons we learn is that we need vertically structured programs top-down They have to become more sustainable. We probably have to develop some use of better residual insecticides that can be applied into the walls inside houses to control Aedes aegypti for longer periods of time. We have to reinvest in infrastructure to do this kind of work. And we have to have triggers for emergency response. When we know that a virus like Zika or chikungunya is arriving in a region, we have to be able to react very quickly. And we have to have sustainable government-community partnerships, an integrated approach going in both vertical directions, and the control programs have to be regional and even local. So some of the new techniques that have become available the last couple decades are some very nice, inexpensive, low-tech lethal ovitraps that attract Aedes aegypti females when they're gravid and they land on a sticky surface or are exposed to a lethal insecticide. There are spatial repellents, even impregnated curtains in places where people don't want to screen their windows because it limits ventilation. These curtains can repel mosquitoes from entering homes. Newer residual insecticides that can be applied inside homes. There are new methods to release sterile mosquitoes developed by Oxitec in the UK. These sterile males then go out and mate with wild females and essentially sterilize them because at the larval stage a lethal gene kills those larvae, and so the transgene doesn't persist in a location, and the populations of mosquitoes on a small scale at least can be reduced by up to about 90%. And finally, Wolbachia, which are natural symbiotic bacteria that infect many different insects, have been adapted for use in Aedes aegypti now. They've been shown to spread naturally in populations in several parts of the world, and in the laboratory, those Wolbachia also suppress the replication of dengue and chikungunya viruses and likely Zika virus as well. So we're entering a new era that should allow us some new opportunities to use a variety of different tools that are moving through the pipeline for controlling these vectors. But critical to this program will be community engagement, the coordination with product testing like vaccines and therapeutics, and combinations with vector control could be very important. And then the international mobilization of resources is also going to be key in these efforts. So that's the end of Dr. Gubler's material. I don't think we have time to discuss all of those questions, but this afternoon we'll bring them back.